Welcome to the second session of today's Liveness Symposium, which will be measuring liveness. And we've talked a lot about measuring, both from a sort of critical perspective, but also from perspective, we've seen a lot of data. So I thought now is a good opportunity to actually do some measuring and see what that looks like. What it actually feels like. So this is now, we can reveal, well, where all this setup comes from. So this is actually the mobile EEGs, or electroencephalograms, that we're using for this research project. And I'm going to sort of show you, so it's all set up now. So I'm going to show you what it looks like. There we are. I'm going to have sunglasses because the projector is very bright. So for my demonstrations, while I'm explaining, I will wear these sunglasses for a little bit. I might also move out of the screen, but I think maybe I'll move out of the screen. But anyway, it's easier like this. So what you're seeing here is is my brain activity. And you see already it's a fiddly thing. Sometimes the cables don't like me, and then it gets very distorted. And there's lots of artifacts, as they call them. So what you see here is each of these 32 squiggly lines is one of these electrodes on my head. And they record my brain activity in real time. Now, it's not just my brain activity here is there. So it records, because electrical activity is recorded, it picks up a lot of other things in the room. It picks up, for example, some electricity from the projector. But generally, a lot of the stuff there is actual brain activity. And I want to do a few other demonstrations of how reactive and fast the signal is. I've got gummy bears here. So I'm just going to eat a few gummy bears. And then you can see, if you start chewing, then you see my chewing in the EEG. So that's not brain activity. That's my muscles, which create electrical impulses throughout this machine. So that's not my brain that's doing this. Another thing, if I take off my glasses, if I blink, you see that too. Yeah. So Jamie in his talk talked about how the eye itself is a bit like a battery and produces signals, or like has electrical voltage. So, and my eyelids have muscles too, obviously. So if I move my eyes looking from left to right or up from down, or blink, then this will also create distortions in the EEG, which actually make it harder to identify the brain activity that's in the signal. Generally, a bit of structure. So the more at the top you are, so I'm pointing to the top rows there, those are the frontal electrodes, as we call them. So this is the front side of my brain. And as you move backward, or as you move down, you move backwards. So those electrodes at the very back are at the back of my brain. So what this means is that the EEG, as we also call it, contains both information about where in the brain activity happens, and crucially, when something happens. And the EEG in particular is very good at telling when something happens, but not so good at telling where something happens. So there's a rough distribution of, you can sort of get some clues about where in the brain these waves are come from, but it's there are other techniques which are much better for that. Now, there are a few things that I wanted to say in terms of measurement that I think, just to say why I think EEG is really interesting to study in the context of liveness. And the first thing I want to say is that it's really fast. So we've talked a lot about how liveness is conceptualized as something, as something ephemeral, as something that happens in the moment. And language, by its very nature, is always retrospective, in the sense that, it's a brain process in itself that takes time to unfold. So talking about something is not a very quick process, but the brain activity itself is very fast, and the EEG can pick up on the fastness of that. Haley Hung also in her talk talked about, the scale of 40 milliseconds at which social interactions happen, and how, let's say, the psychophysiological measurements that Jamie and also Daniel have presented in their respective works are too slow to pick up on the speed of conversation, on the speed of turn taking. But the EEG can do that. Yeah? So if there is something in my brain activity that is related to liveness, to in the moment liveness, I would be able to find it with EEG, but not necessarily with these other techniques. Another thing that I want to say about it is that this particular setup that you're seeing here, it looks really simple, but it's actually really groundbreaking. So up until a few years, getting this signal would require me to sit in a lab. But now with this particular machine, which is, I can hold it up here, so it's fair. I mean, so this is what we call the amplifier. So this is all that's there, and then there's a little screen to do that. All that can be carried with us and can get with us into the live performance. So we have a unique opportunity, and that's part of the novelty of the project, I guess, to take brain measurements into the sort of performative space. And I think it's actually true that what we overall we have 15 of these machines, and I think we are the first. No, I guess that we're the first to sort of try to record brain activity from more than 20 people in a room at the same time. So that hasn't been done before, and that's something that we're going to do hopefully later this year. So it's a really novel setup. The other thing that I want to say, which is particularly relevant for liveness, is that the particular patterns that you see here in the EEG are indicative of different cognitive processes. So we talked about liveness as joint attention, and one way that attention is visible in the EEG is through alpha waves. And I'm going to try to produce a few now. I need to close my eye for that because I will explain in a minute why. So I'll close my eyes. Maybe Henny can shout when or Matthias when they see alpha waves. Yeah, you see these sorts of over there, these sorts of really, there. You see these sorts of larger wavy shapes. Those are called alpha waves. And that's an indicator of me going into a sort of relaxed state. So it's a correlate, a neurocorrelate, as we call it, of inhibition or lack of attention in that sense. And one way that neuroscientists measure attention, and that has been done before, is that, or what has been shown, is that if I listen, for example, the paradigm that Daniel showed, if I listen to a story with someone, if I talk to someone, if I sort of engage with a certain person, my alpha rhythm will synchronize with that other person. So they will sort of fall in line with each other. They will entrain to each other. That is the word. Now, a few funny things about the alpha rhythms. First of all, thinking about standardization, we all have our own alpha rhythm. So the EEG itself is like a fingerprint. So everyone's EEG is totally different, and everyone's alpha rhythm is slightly different. Yeah. So some people, so it's always in the range between 8 and 12 hertz, so alpha 8 or 12 cycles per second. But whether you're more close to the 8 or close to 13 really depends on you. So the EEG can also visualize and measure neurodiversity in that sense. That's kind of the sort of like little thing that I wanted to show about how the EEG works. We're going to keep it running in the background, but I want to now show, do a few slides. Oops, see, now it's not, doesn't like that I'm moving. I want to show a few slides a little bit more thinking about the neurocorrelates of liveness in particular. Here we go. I need to share my screen for that. Oh, shit, I want to show, do I want to show videos. Yes, I do want to show videos. So let's do that again. Video settings. Sorry, I'll do that again. Stop video. Start video. Share my screen. Share sound. Optimize for video clip. That's what I was looking for. Sorry. So hopefully you can now see or all see my screen. Can you see my screen? Yes, I am screen sharing. Right. This is a good opportunity, by the way, to acknowledge the work of Victoria Ford, actually, who designed all of the visuals or visual identity of NEUROLIVE that you've seen so far, and that I'm very happy with. And that doesn't matter, but that I think is really, really nicely used. And in another context, we might ask her to sort of talk about how she came up with this particular visual design for the NEUROLIVE project. But for now, I want to talk a little bit more about the idea of science and the scientific methodology as a way of reducing complexity. And when I think of the scientific method, I don't think of it so much as a way of reducing complexity, but as a way of building complexity, generating complexity from very few elements, or for only very few features. So in that sense, I think science is quite choreographic. It's really about taking a few elements and then trying to see how far we can build, how we can generate complexity through them. And I'm going to try and do that for the idea of liveness as a form of joint attention. So really starting from Daniel's idea that liveness is somehow related to entraining or sharing of eye movements, but in this case now brains of each other. So going back to the entrainment of alpha rhythms that we saw earlier. So let's start with the simplest situation where we have one spectator and one performer, and how liveness lies in their mutual entrainment to each other's alpha waves. Now, starting from that, we can now deal with different kinds of mediation, so physical liveness. So let's say if one of the performers or the other one is now through a screen, is on Zoom rather than in person, then that somehow reduces the amount of social information that is available to me and might impact or reduce the kind of entrainment that's possible. Yeah? So the reduction in liveness now is sort of related to the lack of to the reduced entrainment that comes from the fact that there's less information transmitted between the two. We can also think of a situation which we haven't really talked about, but which of course is possible. So let's say two screens talk to each other. Yeah? So I'm mediated, the other person is mediated too, and the two screens are talking to each other, which is probably likely to reduce liveness even more. And then we have a situation, of course, which is simply the reverse of the first one, where the performer is live, as it were, and the spectators aren't. So that's the situation in which SYMPOSIUM's collaborators will find themselves later on this afternoon when they perform work for us at home while we're watching on our screens, and they can see us. No, they can't really see us watching. The idea of entrainment between people can also help us deal with remembered liveness, or the relationship between memory and liveness that we thought about before. So rather than having an actual person in front of me, I can remember a person and entrain to that. And that is possible because the brain doesn't really distinguish between what's past, present, and future, but every memory really is a replay, or that's how we think about it now, is a replay of something that you've already experienced. So brain activity the brain activity when you remember something is essentially the same than when you perceive something. It's just that it is endogenously generated by the brain and is not linked to an actually incoming external stimulus. So we can explain liveness, remembered liveness, through this idea. And then obviously we can sort of multiply the number of people who are spectating, and we can look at the social aspects of liveness that we've encountered so far. So we can ask, to what extent does my entrainment or my co-presence or my sharing, my entanglement with the other person depend on the other people around me? Do I need to be to experience their presence too in order to, and to, in what way might that enhance or strengthen my entrainment to the performance that's happening? An easy way to look at that would be asking questions around how many other people are experiencing this together with me. And then we can do the same thing on the performers. So of course, there's not just one performer, but there are multiple performers too. And we can ask, how does the entrainment that happens between spectators and performers depend on the communication between performers? Does it matter on whether they play a role or not? How is it related to ideas about authenticity that we came across before? So being, performing something, or sort of being sort of present in a way. So we can look at that too and quantify that perhaps in terms of entrainment between the performers themselves. And then we can obviously blur distinctions between spectators and performers. So this now leads us into the work of Zhou Feng and Anusha that we saw yesterday. So what about situations in which there is no clear distinction anymore between spectators and performers, in which the performers are both spectating and performing at the same time? And how does that impact on entrainment of alpha rhythms between people? And then finally, last but not least, we need to acknowledge that all our brains are different. So I've said earlier on that we all have individual alpha rhythms, but this term of individual difference is a sort of psychological term that captures all possible differences that might exist between people's brains, people's bodies, and people's experience. So now going back to Claire's ideas and Claire's work on inclusivity and diversity and accessibility as well. So taking into account that people have different brains, different alpha with different functions and different bodies and different levels of experience, we can take that into account in sort of trying to sort of understand liveness as a form of joint attention. Now, this is as far as I'm going to take it, as I've run out of animations and out of color scheme. Your eyes probably hurt already, so I'm not, but this is just to illustrate that we can build complexity. A lot of the complexity that we thought of needs to be reduced. Perhaps we can build complexity from a rather small number of ingredients. I want to close actually by relating this work, the NEUROLIVE project, to the work that we did a few years ago with Matthias and a number of performers whose names I will give later, which is sort of the starting point for this project and was the first one to give us some clues as to where liveness might lie and that we might be able to measure liveness as a form of entrainment. I'm just going to show this for a little bit while I talk over it. So this is work that Matthias did together with 10 performers only over 3 days, which was really an amazing achievement. So to produce anything really. Where we were sort of more specifically interested in the relationship between synchrony or synchronicity and audience engagement. And we used the same measures and the same sensors that Jamie and Daniel introduced in their work earlier. So we use these accelerometers to measure performers' movements or performer synchrony in this case, using recurrence rate. So that's a technique that Riotta introduced in his work as well. And we had spectators, and from them we, through using tablets and the same measurements, we collected data of their engagement, their enjoyment of the performance, and their heart rate, also through these through the empatica sensors. And what you see here is that, so the red line are changes in synchrony among those group of 10 performers over the course of half an hour. And in blue, what you see is the sort of level of average enjoyment of a group of 15 spectators watching this performance. And what I want to say to this is that there seems to be a causal, and there is some kind of relationship, a predictive relationship between what performers do and what audiences experience. And that doesn't mean that this that everyone always knows what that relationship is. Quite the contrary. So it may, if you ask performers, you know, what are you doing? What is synchrony? They may give very different answers. And even so, spectators will give very different answers if you ask them as to what they think is currently happening. But in these ratings, we do see that synchrony predicts enjoyment in this case, but that's not a simple, straightforward relationship. It's not that. So here are sections where collective stopping, so stopping together all at the same time is a very strong moment of synchrony, creates these little peaks of pleasure. But then there are other moments where this relationship is negative. So it's still predictive, but here there's a section where the group gradually descends into more chaotic movement, and that's highly enjoyable too. So we do see a predictive relationship, but it's by no means a simple linear one. But where is liveness in this? And that's kind of the question that sort of we want to look at in the future more. And I think liveness in this particular study, we can sort of we can glimpse at it when we compare performances from different evenings. So the whole performance, so this was a work, it's a score that Matthias created for this study. So we performed it 4 times in total, and this is data. So let's say the dark gray ones, is if I take, let's say, synchrony and enjoyment from the same night of the performance. So like so performers perform synchrony move over time, and audiences give ratings of that. If I take data from the same performance, then I can find significant relationships between movement synchrony and enjoyment. But if I take spectators from performance 3 and spectators from performance 3 and the performance data, the synchrony data from performance 1, then these relationships vanish. So what that means, and the same holds for if I take heart rate data. So to use terminology of Anna, what I think this means is that the relationships we see, those relationships between performer and spectator, are not related to the work. They're not related to the score. They're related to the performance. And in that sense, so changes in synchrony among performers predict enjoyment and heart rate among spectators, but only if the synchrony and enjoyment data from co-present performers and spectators are matched, if we use data from the same evening. So this suggests to me that we can use measurement in this case to sort of get a glimpse of what might liveness mean, or certainly we can get a glimpse of what this distinction might look like, the one between performance and work. And on that, I'm going to close, and I want to thank Matthias, but also the performers of this work. So Ben Esch, Andrea Buckley, Michael Kitchen, Antonio Della Fe, Kip Johnson, Evangelina Colera, Sarah Wookie, Annie Locke, Rosalie Walfried, and Robert Vesti, who made that possible. And for transition, I've conveniently glossed over with this nice little sign what it means, what entrainment means, what does it mean to entrain between brain states of people, and what is a brain state anyway? And this is where my colleague Diego comes in, who will talk about what brain states are and what the relationship between brain states, perception, and experience might be. Thank you very much. So I think we've got until 12:45, is that correct? Oh, okay. So I'm handing over to Diego now. We can do questions later.
Should I jump in now?
Yes, wait, I'm just needing resume share. No, stop share, stop share. Yes, jump in. I'll stop sharing my screen. Your turn.
My turn. Let me share myself. Well, hello. I'm Diego. Thanks, Guido, for the opportunity of being here. It's been really fascinating and really enriching for me to see, such a diversity of backgrounds, really different from the typical conference that I normally attend. So myself, my background is on computer science. So my work is as a computational neuroscientist. So what I do is to develop computational models and algorithms to try to describe brain activity, which I then use to try to address questions that are interesting to me or to the field, like, for example, which are the sleep state, the mental states or brain states that precede the onset of sleep, or how distractions affect the perception of pain. So normally in a standard conference, I will go into fairly detail in one of these models. I will talk on one of these questions that are interesting to me. But because this is such a diverse audience and a bit different from what I'm used to, I thought it would be a bit more interesting if I instead speak about the principles that underlie these methods and these models. And I don't mean how we do design the algorithms or we do the programming or whatever. I mean, which are the actual why, the philosophical ideas of how the brain works and how that relates to behavior and ultimately to what we are. So I thought it would be interesting if I give you like a super quick and very non-deep tour on the different ways or different views of how the brain works, and in particular the one that I think is most reasonable, and how that frames the methods that I will present next, which we think could be used in interesting ways, or Guido thinks that it could be interesting ways to measure liveness. So neuroscience, or computational neuroscience, is really a crossroads of sciences. It is computer science, it is philosophy, it is psychology, it is physiology, and addresses questions as complex as where in the brain does perception occur, for example, what initiates my finger movements, or what makes my mind fly. So neuroscience is in a way a bit of a weird science, because it's not just that these questions are difficult to respond, but the questions themselves are difficult to pose, so we don't really know what is appropriate to ask in the first place. So today's neuroscience is very heavily influenced by the thought of the philosopher and psychologist William James, who sort of compartmentalized the different sort of domains of the human mind into different categories, including, for example, things like attention, memory, and so on and so forth. And today, most neuroscientists sort of ascribe to one of these domains and dedicate their career to really delve into one of these categories. So that is something that we could nowadays criticize to some extent, because, I mean, at the end of the day, these categories come from ancient thinking, come from even thousands of years ago. Humanity conceived these categories at a time where the brain was actually thought to be a cooling device for the body, and the thinking and the feeling was thought to happen in the stomach and in the heart. So maybe it would make sense to think of which are the appropriate questions to ask in the context of evolution, for example, how our brains have evolved through the history of our species and how it relates to other species and so on and so forth. But that's a bit of a complex topic that I don't want to get into much. So this James thought and today's neuroscience is still heavily influenced by British empiricism, which itself have roots in Christian thought. So one of the main principles of British empiricism is determinism. So basically, things are the way they are. And the second thing that is critical, key, is very important to this way of thinking, is that science can only be made of what we measure and what we observe. So knowledge is based upon observation and nothing else. So that takes its more radical view in terms of psychology in the hand of behaviorism. So behaviorism basically tells us that behavior is just a function of stimuli and of reinforcement, which means basically whether we like the stimulus or we don't. So the brain can be thought as sort of a passive device that takes a stimulus and transforms that into some behavior or actions, and that is thought to be quite deterministic. Even though obviously we don't know the rules that determine this thing, but at the bottom of it, it is deterministic. So it's a little bit as if the brain was a computer. But the computer analogy really belonged to a different school of thought, which is the cognitivists. So the cognitivists think that what it really makes sense is to conceive the brain in terms of information. So information is the key thing to understand the brain. So the brain recognizes patterns, associates patterns, works in the forms of rules and algorithms, and that is what we should really consider. So today, the metaphor of whether the brain is a computer or not is really a very alive debate. So whether we can think of the brain as a computer or not is something that we still talk about. Then the post-cognitivists, with great words such like autopoiesis, think that wait, maybe information cannot be the whole story, cannot be the key thing to understand brain function. So what the post-cognitivists argue is that it is not information what matters, it is not stimulus action what matters, but what matters is understanding the brain as a sort of a perpetual self-generator of neural patterns. So what matters is not the information that comes in and gets transformed in an action, but what matters is the activity that gets generated from within. So that doesn't mean that obviously that the stimuli and actions are irrelevant. It means that neural patterns were there before that. And it is stimuli and actions what mold these patterns, but the patterns are there beforehand. So it's a way of looking at the brain, so to say, as an inside-in thing instead of an inside-out. Sorry, the other way around. It's more an inside-out is the correct way to see the brain for the cognitivists as opposed to the outside-in. So that obviously there's some developments on that. Is what is called embodied cognition, the things that it's not just the brain what we should consider, but the entire body. So the brain and the body makes one single thing, and cognition happens as an interaction between body and brain. So what does mean in practice for us, for me as a person who develops models and tries to understand the brain from data? Well, first it means obviously that things are complicated and that excessive reductionism is no good. So that's kind of almost obvious. But most importantly, that mental and behavioral variability are of the essence. So we evolved to change. So our brain responses are not always the same. They change as we change. So that is in contrast with the behaviorist view, which basically looks at, as I said before, as brain as a passive device that more or less transform one thing into another. But this idea of the brain as a self-generated device or organ is really entangled with this idea of variability. So just to give you like an example of an experiment of very, very typical of what we do today in science. So this is, for example, this is an EEG signal of people being shown faces or cars, the two types of stimuli. So what the field typically does here is you show these things many, many times, and then you get the average signal for faces and the average signal for cars, and regard the variability around this average as some sort of noise. So what I'm advocating here is that is this variability exactly what matters to understand brain function? So this variability is what defines us as human beings. So what can we do about this? So that's the context, the sort of philosophical context of the methods that I want to present. So what can we do about this? We can develop methods that try to address and characterize this variability instead of just treating that as noise or something that is irrelevant. So one of these models that I've, sorry. One of the models that we could use to do that, and the one that in particular I've been working on in the last years, is called the hidden Markov model. So the hidden Markov model, what it does is tries to describe the brain signals, for example, the EEG signals, as a succession of state activations. So there's a set of states that go on and off that occur and reoccur. So the hidden Markov model, or HMM, has 3 different elements. So one is the state time cursors, which is basically telling us when each state becomes active or non-active, which is a probability per state and time point. There is one description of what a state is, or what each state is, so contain information, for example, of how the different brain regions communicate with each other. And because the HMM is a dynamical system, there's a third element, which is a transition probability matrix telling us what is the probability of going from one state to another. So all of these 3 things are encoded as parameters and are inferred directly from the data. So we as scientists, we don't specify any of these things. We just present the data to the algorithm, and the algorithm comes up with these different states, when they occur, and what they are, and how they change between the others. So one, I think, quite intuitive example of how we use this model is to try to come up with a hopefully better description of sleep. So sleep is classically described as a number of stages, typically 4 to 5 stages. So there is normally someone who understands about sleep and takes the brain signal of the EEG in windows of 30 seconds, and then knows which features he has to look at to classify these different windows of 30 seconds into these 4 stages of sleep. These 4 stages of sleep were defined almost 80 years ago, by the time that EEG was discovered. So we thought that maybe it would make sense to try to describe sleep in a more data-driven way, to let the data speak a little bit more instead of having someone sort of going manually window after window, classifying these sleep stages. So we just gave the data to this HMM algorithm, who was able to produce a description of sleep with more states and quicker transitions. But what it actually describes features of the sleep at least as well as the traditional view, but offers additional insights. So that's a good example of these data-driven methods. Another example is visual perception. So there are few things in the brain that are as fast and presumably as stereotyped as visual perception. So when you see a stimulus, you feel it almost instantaneously. So it's something that happens very quick, like 100, 200 milliseconds. You see something, you notice what it is. So the way people has been investigated visual processing was like presenting, giving a stimulus to a person a number of times, and then those are called trials. So you get a number of trials for, let's say, 1 second each. And then at each time point, they use some called machine learning algorithm that tries to predict from the data which stimulus is the one that the subject is looking at. And then that model applied to different parts of the brain can tell you how and when the brain is processing that visual information at each time point. So you come up with a sort of cascade of visual activation from the very moment that the visual stimulus appears to maybe 1 second after, when that thing is encoded into memory. So that is, I mean, there's a lot of progress in understanding vision this way, but as I was telling before, it has this element of behaviorism, because it's fundamentally ignoring the fact, as I said before, that brain responses are not always the same. So we applied the models I was introducing, a variety of HMM, and what we found is that even though these things happen quick, there are differences for each presentation of the trial, for each time you see the thing, there are differences in the way the brain responds to it. For example, in the timing. So when you see something, the first part of the cortex that processes visual stimulus is the occipital cortex here, and then sort of propagates towards, for example, temporal areas. So that cascade of stimulus processing, we found that it was way more variable in their timing than we thought it was. So what that means, really. And what this variability comes from, it can come from many different things. One possibility is that this variability comes from maybe the brain state at which you were just before you perceived stimulus. And that's something we actually offer empirical evidence on. So the whole brain activity just before has a profound impact on not just the timing, but the very nature of this brain response after the stimulus is presented. So, so I've been talking a lot about these sort of philosophical views, and when I present these methods, really I'm talking about something that is quite simple. It is this temporal variability. So, but yeah, it was to reemphasize how important this is in terms of how we conceive brain function. So if we think of the brain as a self-organized organ that changes all the time, there is an element of unpredictable. So this variability is acknowledging that our brain is inherently unpredictable. And that, I think, is a very, very important distinction. And well, I think many of you are artists and not performers. I think that idea of improvisation, of things being unpredictable, I think will resonate well in you. So that is what these methods are, I think, they're trying to acknowledge in essence. So what next, for at least for the interest of liveness? So far, as I just been discussing, we're measuring states of synchrony between brain regions. So for example, we see this visual stimulus, and then there is some communication between different areas so that we process that and we perceive that visual stimulus. So what Guido thought, and I think it's a great idea, is why don't we try to use these methods, not just to find moments of synchrony between brain regions, but states of synchrony between people's brains, maybe when there's a performance going on, either in the audience or in the performers. So we could try to find, again, in a data-driven way, which are these perhaps special moments when all these brains come together at once, driven by what is happening in the performance, and how this between brain connectivity, this special moment of togetherness looks like. So maybe those methods are a good way to do that. So that was what I had, and I'd like to use this opportunity to thank you, and also to say that I'm hiring. So if someone is in search for a job, just reach out.
I'll join.
You'll join.
I was totally rude of me, totally forget to not introduce Diego. So I'll do that now. Diego is associate professor. First, clap, 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 clap.
Thank you.
So Diego is associate professor in Denmark at the moment, and he went there also funded by the European Union, actually. But before that, he did his degree in statistics and machine learning at the Polytechnic University in Madrid. He then went to Oxford to do his postdoc with Mark Woolrich and Steve Smith on methods to quantify brain network dynamics and their relationship to behavior. So he's currently building, as we just see, his own research group in Denmark. I should also say that I'm very pleased that he's here because he's actually just became a dad.
Oh, yes. Yes.
Less than 2 weeks ago. So yes, I said to him, if you don't have time to prepare your talk, then you can also just hold the baby into the camera and we'll all be happy.
I've been following these 2 days as much as this 10 days baby has allowed me to. Because, man, boy, 10 days babies are trouble. I'm just... Right.
I'll unshare your screen, and I think we have a bit of time for questions. Jamie has his hand up. Come in, share your camera.
Hello there. Hello there, Diego. Lovely talk. Excuse the background. I'm in the middle of a building site. I very interested in your use of hidden Markov models to study the dynamic changes. And I guess one of the questions I have about those is that you have to set the number of states. So you have to make decisions about how many states you're going to have. Is that correct when you're building those models? So how do you go about deciding that actually for, if you're looking at a series of states? Are you having a state per brain region, or are you, how are you deciding how many states you're going to use when you're using these methods?
So there are different varieties of this model. In the sort of more basic one, you have one state active at each time point for the whole set of data you're looking at. So you are looking at the whole brain, then you have one state active at each time point for the whole brain. You could run the thing for different parts of the brain and try to combine these different chains of states together. But let's stick to the most basic one, where we have one chain of states. The number of states, that's a good and a common question. So there are different ways of doing that. There are varieties of data that can automatically infer the number of states at the expense of being more expensive to compute. In my experience, the number of states is gonna tell you, it's gonna determine the granularity of the solution. So you have more, it's like, I don't know if you're familiar with ICA, because it's the same. So if you have more states, you're gonna capture more detail in things. You have fewer states, you're gonna capture trends that are more principal, so to say. So I do like to experiment with different number of states, because I think that is quite informative itself. So I am not, in the context of neuroscience, and engineering is a different thing, but in the context of the neuroscience, where we are after understanding and interpreting, I think that experimenting with different number of states is much more productive than just, coming up with some automatic way of doing that.
Yeah, if I may follow on from that, and that there's it's this exploration, because we don't know how the brain works really, and you said it's completely unpredictable, and but there is a structure there. There is a temporal structure. There are behaviors that we're trying to uncover, trying to understand. And you set out a hypothesis, and maybe that is now previously it was, I think this brain region has this particular duty or job, but now it's also, well, there's a temporal sequence that has a particular function. So I guess it's not that it's unpredictable, it's trying to find what those sequences and patterns are. So the hypothesis is, that would dictate how many states you try in your model, I suppose, as well. Or...
Yeah. I didn't mean to say that was completely unpredictable, but neither the opposite. That was my point. but yeah, you're right. So the sequence of states, I think the sequence itself is a very informative and very meaningful thing, I think. And that obviously changes if you choose to have more states. But that's, I mean, that is, at the end of the day, those sort of decisions are like in almost in everything you do, right? So how many, which do we use MEG, do we use EEG, do we use this, do we use that? So there's always choices. And that, I think we should face that and experiment. That is, I think, the most productive.
I just think it increases the number of parameters exponentially, and that makes it exciting, but also very difficult to work with. We've already got, how many channels are we looking at?
To integrate in a sort of data-driven way different solutions. So that's something you could do as well. So that there's some middle point, you know, and to just, you know, going crazy and just running once.
Well,
other questions, comments, thoughts?
Should I?
Anna, yeah, I'll like, let's bring Anna back.
Hello.
Hello.
Yeah, I mean, I really enjoyed your overview of different philosophies of brain stroke mind, Diego. And I was struck by the fact that, let me try and get my thoughts in order. Perhaps you should have read out my question. Whether you think that neuroscience provides evidence for one or other of those philosophies, or whether you think that actually the philosophy or the conception of brain or mind shapes the way that the investigation happens neuroscientifically, and or some combination of the two, perhaps.
I think most realistically, the philosophical conception shapes how we do the research, most realistically. And whether neuroscience can sort of give us some insights on which these views are more correct than others, but it's difficult with these things to talk about correct, really. So, you know, we're really at the boundary of science in a way. So the things that, like consciousness, for example, is such a difficult even to define thing, that you know, it becomes difficult to think about empirically, and that's what philosophy comes in. So I think it's a dialogue between the two, rather than neuroscience telling philosophy, like, you know, things are that way. I don't know if we will ever come to an answer to these things, or maybe it's, you know, it's just some of these questions are just probably impossible to answer. But I think we learn from each other.
Yeah. Yeah, and I wonder whether there is a space for neuroscientific inquiry to actually cast doubt on the plausibility of some philosophies of mind, because they reveal that the brain operates in a very different way to the way assumed by that philosophy.
I believe so. For example, with I presented behaviorism. I think those all ideas of Pavlov and Skinner, I mean, those are tremendous progress, obviously, but I think today's more detailed knowledge or view of the brain sort of, in a way, debunks some of these things to some extent, at least. So I think that is a good example. What I don't know is what are the limits. That's what I don't know.
Thanks.
Thanks.
Yeah, Anusha raises hands. Shall we bring her in? Cool.
Yeah, I raised my hand, but I'm not quite sure if this is, just following on from what Anna said, is just, I wondered whether you could design an experiment to compare how the different models actually affected your results. So if you, I don't know how that would work, but if you designed an experiment that came from like all the different perspectives and see how that would somehow change the results. And the other question I had was, because you kind of jumped from the sleeping thing, to the being together, and sort of just a question about whether, about the experiment having to find out, you were saying about the unpredictability, and I was thinking about patterns within the unpredictability of maybe something could be with neurodiversity, for instance. So people, and like a question that maybe comes from what Matthias might say, there's something that's used in dance a lot, like from Deborah Hay, saying like this thing of getting what you need, and how people might have very different needs, and that might be something that might become more obvious in something like neurodiversity. So I was thinking about whether somebody's experience or need of dreaming or something like sleep might be very different in different people, which might show in different neurodiversity. I don't know why, it's just something I was thinking.
No, that's super good comment, actually. So I didn't talk about this, but so this variability in time is one side of my research, but the other side of my research is understanding how this variability is different between people. So how this variability characterizes people. And I think that speaks a little bit to what you're saying.
Yeah.
And that's really, I mean, it's a deep thing. So I'm just starting my project on this, and we're hoping to describe how neurodiversity in a bit more detailed way, so to say. With regard to your first question, whether there are sort of overarching experiments to sort of decide between these different views, I mean, not that I know of. I don't know of any experiment that really confronted this.
But I mean, I guess in a way, I wonder if in a way you have shown that already. I mean, to me, what your methods and what your work shows is that, you know, these traditional models of behaviorism and stuff, they ignore, or they get rid of variability, you know, sort of they sacrifice variability for the sake of an average, in a way, you know, for like having something, which is how I think often neuroscience is perceived. You know, that's the popular understanding. You reduce, it's the reduction of complexity to something, to an average, which then no longer represents the diverse reality that's out there. And I think, you know, simply by plotting the variability that exists, you show that, you know, those models in a way, so like, you know, taking into account the variability and showing that matters, sort of, I guess, shows that the starting point of like simple stimulus input, stimulus output is not correct.
And I guess also another thing that it can be tested experimentally, and it speaks to these things, is how important and how prevalent is this spontaneous activity. So that thing that is happening before, during, and after the stimulus is presented, that activity that is in sort of way undefined, but however that heavily influenced how we perceive things and how we feel. So I think that acknowledging these self-generating patterns that are continuously, from the moment we are born to the day we die, going on and on and on and on, and I'm thinking of that as something meaningful and not just noise or something, you know, to average out. I think that if we prove that those things are relevant, that I think, now that I think of it, that is a good experiment to validate, I think, the latest view.
Yeah. One more question from Anna in the Q&A. Thanks so much, Diego, for an amazing talk. There has been some work on brain synchrony and naturalistic stimuli, for example, from Yuri Hasson's lab, but I guess group brain synchrony hasn't been done as much. Do you, Guido, have an idea of how the synchrony across individuals that are tested in separate lab sessions may differ when individuals are together in a group audience setting? Thanks and congrats. I guess maybe I'll start with that one. Yeah, you're right, Anna. So there is quite a bit of work on sort of inter-subject synchrony or connect or sort of synchronization among people, but it's always done in separate sessions. So what's done is in either sort of in imaging or in EEG, you record data from one person and then that person leaves, and the data from another person you collect, and then you can obviously relate and correlate and analyze these people as a group, but they've never formed an actual group. And so there are a few studies with EEG now that have, I think the largest group is around 14 or 15, but then the quality of these systems that can do this is much lower than the ones that you're looking at now. And the sort of general signal is much poorer, and it certainly wouldn't allow for any of the analysis that Diego is describing. And so basically it's an open question to what extent sort of synchronization studies from individually measured people map onto when you actually collect data from groups of people together is a sort of open question, because that hasn't been, that simply wasn't technically possibly until very recently. Diego, do you want to add to that?
I think that there's been, the only thing that I know of is these experiments where there are two people in the scanner with the screen looking at each other. And there is some attempts to do that, right? With maybe 2 or maybe 3 people doing some game or whatever. But nothing like you're proposing, to the extent of my knowledge.
All right, I think Susan asked a question. Yeah. Yeah.
Not really.
Hi, Susan.
Hi. How you doing? It's not really a question. It's Susan Hiller. I don't know if you know her. She's a visual artist. Unfortunately, has passed away. American who worked in Britain. But she did this series, what was called dream mapping, where she selected this particular site that was known to have this almost spiritual kind of essence about it. And people did a community sleeping. So it was not a it was a performance, but without an audience. But the cool thing was that they, Susan asked them to draw these maps of their dreams. It wasn't written, so they were in a kind of automatic writing state, automatic drawing state between sleep and awakeness, of which they composed these maps. And then when she put them together on translucent paper, found synchronicity in loads of different places. And I did a workshop with her at the Hayward Gallery years ago that was specifically on this. And I mean, it's weird. It lies between the artistic world and the scientific world of finding these kind of correlations. And there's definite liveness within this particular event. However, it's not to an audience. It's almost the liveness in the shared experience and also in the sharing of the kind of accumulative research that evolved from it. And then Susan went further into curating this as an artwork. So it had kind of these reiterations.
Yeah, thanks for that contribution. I guess my only answer to my question to that is that I'm not surprised that these overlaps exist at all, because I feel like we're asking the same questions. Ultimately, art and science, we are asking the same questions. I mean, what does it mean to be human? What does it? So the questions are the same, as the approach is different.
Absolutely, yeah.
And I think I'd like to think that it's really, I'd like to think, I guess, in my work, and that it's really useful to remind ourselves that the questions are the same often.
Yeah, it's just a kind of the methodologies and the kind of play.
Yeah.
The tasking, the play, etc., is of different modalities, but that's part of it. And then how do we kind of collapse these modalities? Are really interesting. And then therefore that gets back with yesterday about the languaging, not necessarily when we were talking about the word measure, and maybe that's the wrong word, because, or measurements should be done within different kind of, not necessarily numbers. And what is the graphs of it? And what is the visualization or the tactile representation of it? To open that up and to not be in a fixed, already preconceived format. Yeah. Thank you.
Thanks a lot. I think that's a great moment to sort of move away. Thanks, Susan. That's a great moment for us to move into the lunch break. We've run a little bit over time, but I think it was absolutely worth it. So we shall reconvene at 2:15 for practicing liveness with Seke Chimuteng Wende and collaborators. Looking forward to seeing you again soon.